This protocol uses a streamlined approach for measuring plant gene expression in response to insect herbivory. By using detached leaves isolated in petri dishes, the insects are easier to apply and monitor during the relatively short period of infestation, contributing to reproducible gene expression data.
This protocol may be adapted to many plant-insect interactions if baseline observations from whole plant systems are considered. The unpredictable nature of insect feeding can be challenging. Therefore, a dependable supply of properly staged larvae and healthy plants provide the best chance of success. Visualizing how healthy plants and insects should appear and behave is instrumental to achieving optimal results, especially for researchers new to the field of study.
Demonstrating the procedure with me will be Francis Perez, a molecular biologist in the Genetic Improvement of Fruits and Vegetables lab.
To prepare nodal propagated tissue culture potato plants, first obtain Kennebec plantlets grown from explant material with at least 3 to 4 nodes. Use a sterile blade to remove the leaves, cutting the branches close to the main stem and leaving about 2 millimeters of branch tissue per leaf. Cut approximately 2 millimeters above and below each node to remove the nodal sections from the stems and arrange the nodal cuttings in a sterile tissue culture vessel containing nodal transfer medium with the branch pointing up. Then transfer the nodal cuttings to a plant tissue culture chamber for 2 to 3 weeks of growth at 24 degrees Celsius in a 16-hour light, 8-hour dark photo period. To prepare the insects for feeding, obtain the desired larval stage of M. sexta and transfer one larva into each wall of an appropriate containment vessel according to the size of the larva.